All right. All right, folks, welcome everyone. So today we're gonna have Eleonora Secchi in our lecture series. I'm gonna read her bio here. Dr. Eleonora Secchi is a soft matter physicist working at the interface of biophysics, microfluidics, and fluid dynamics. She trained in engineering and physics at Politecnico di Milano, University of Montpellier, and MIT, and held a postdoctoral position at ENS Paris. She is currently group leader and permanent senior scientist at ETH Zurich, where she leads the biomatter microfluidics group. Her research focuses on flow, deformation, and transport in living porous media, porous and soft materials, with a particular emphasis on bacterial biofilms. By combining microfluidics, imaging, and physical modeling, her work aims to establish mechanistic links between microstructure, mechanics, and transport, and to understand how mechanical stresses and confinements regulate biofilms morphology, permeability, and ecological function. She has since founding her group in 2018 with an SNRS Prima grant. She has established strong international collaboration, including with the European Space Agency. She was named a Soft Matter Emerging Investigator in 2022 and is actively involved in international scientific leadership through advisory and committee roles. So I decided to read this because it's a very impressive description. We're very happy to have you here, Leonora, so you can have the floor.
Thanks a lot. I'm glad to present here today. And to keep it informal and lively, please feel free to ask any question you like during my talk. Today we're going to talk about mechanics and transport in bacterial biofilm with a particular emphasis on structure and flow coupling. And first, I want to thank my group and the people who did the experiment and did the work here at ETH Zurich over the years, and the funding agency that support our work. And our work focuses on biofilm, as I said, and we want to reframe them as living polymer networks, in which basically the physico-chemical interaction set the structure, the mechanics, and the transport. We use microfluidics coupled with visualization technique to grow biofilm in a very controlled physico-chemical environment, and so gaining reproducibility over their growth. And then we visualize them on scale that go from the millimeter to the micron scale. And this allows us to understand what is happening at different scale and what controls their formation and their properties, as we will see. And once we gain this mechanistic understanding, we use it to control and engineer the formation and properties. So first of all, let's start by defining formally what biofilms are. Biofilms are aggregates of bacterial cell embedded in an extracellular polymeric matrix that they secrete. And they are really ubiquitous because of their resilience. They can be found in industry where they foul membrane, pipes. They are a big problem in space research and space missions, but also in medicine, where they are the cause of chronic infection of medical devices, especially. And also they are a place where antibiotic resistance can be created and perpetrated over generations of bacteria. And all this colonization capability comes from the extracellular polymeric matrix, which is these components that offers chemical and mechanical protection to the bacterial population. Thanks to it, there is this emergent resilience which makes the cells from 10 to 1,000 times more resilient when they are in a biofilm to chemical treatment, for example. This is common to all the biofilms, I would say. However, biofilms can have very different morphologies and architecture depending on where they form. And so basically depending on the boundary condition that they find during their formation. The most studied and common biofilm, the one that we picture in our heads when we think about a biofilm, is probably the surface attached one. And in this one, cells that first are like swimming find the surface, colonize the surface first as cells, then they start producing the extracellular matrix, they form an early stage biofilm, the biofilm then matures and grows in space and time. Sometimes we have a differentiation of function of bacterial functions inside the biofilm until it becomes a real city of microbes. And then when the conditions are not favorable for the biofilm anymore, biofilm can disperse and cells become again planktonic, and then they can colonize other niches. And here bacteria has to switch their phenotype to do this path. Otherwise, they have other options to form biofilm, and those are like more physics related. Because, for example, we can have streamers that, as we will see in details later, are filaments of biofilm suspended in the flow, and there the local flow condition is very important to determine the formation of the structure more than the phenotypical switch of the bacteria. Or also bioaggregates. Bioaggregates are also formed due to forces that are non-biological and are common also in gels and in other soft material, and can be polymer bridging, depletion forces. The formation of streamers and bioaggregates can be very rapid, so in terms of minutes, while this path of surface attached biofilm is much longer. And so from a physical point of view, if we look at all these different morphologies and architecture and time scale and spatial scale, we see a huge diversity. And this is something we really enjoyed in our group, where we tried to study several geometries and see what are the common points and what are the differences. One very, very common features of biofilm is the matrix. The matrix is always composed by polysaccharide, proteins, and extracellular DNAs. Then polysaccharide and proteins are species dependent, so every species has its own different polysaccharide and protein with a different structure and material properties, while extracellular DNA is just the chromosomal DNA that for some reason gets out of the cell. And all these components interact with principles that basically are dictated by polymer physics, so bridging, cross-link, ionic interaction. And this confers biofilm their internal cohesion forces. From these cohesion forces, they get both their mechanical properties, and in particular, the most studied is the viscoelastic response that allows them to absorb and dissipate mechanical stresses under flow shear or under compression, but also the matrix hinders transport. So diffusion is suppressed, but also advective transport, and so thanks to this, gradients are created, both of resources and waste inside the biofilm. We will touch upon both today, but we will start from the viscoelastic response. So what does viscoelastic means? Let's start from the real basics. A viscoelastic material is a material that when we applied a load, a force, instantaneously deforms elastically with a reversible deformation, and then starts flowing like a fluid, a very viscous fluid, over longer time scales. And this second deformation is irreversible. And so when we remove the load, the elastic deformation is recovered, as we see here, but the viscous one is maintained. And so basically biofilm behave a little bit as a spring and a little bit as a viscous fluid. And this is really ubiquitous. We can take those are wild biofilms found in the environment with many species there, also algae and fungi probably. And we see the deformation is like textbook here. This is a biofilm grown in the lab from a single species model organism, and they have the same shape of the deformation. So they share this viscoelastic behavior. However, what is really different and this is something that fascinated me a lot when I started my group, are the viscous and elastic properties, like the magnitude of these moduli spans order of magnitudes. And this is quite uncommon for normal material that can span order of magnitude, but not 10, as in this case. And we studied what controls the viscoelastic behavior a little bit. And I'm not going into the details here, but what we found in bulk, so using rheometry, is that biofilm formed under flow are like stiffer than those developed under quiescent conditions, so no flow, moist environment. And the fact that environmental conditions set biofilm rheology, we will come back to this also later, is extremely fascinating, but also creating complexity if we want to have an absolute abstract characterization of the material, because how we grow them matters a lot. Then there is a second behavior that is interesting and potentially very important for the removal, and is the nonlinear rheological response. Basically, this response is activated when the deformation is like very big. So the biofilm being torn apart, so to say. And this behavior, this nonlinear rheological response, where basically when the deformation is big, they change their rheological behavior, has been observed, but not mechanistically understood. And so if now we combine these two responses in a biofilm grown under flow, we have to imagine our biofilm and then a flow shear touching it on its boundaries. And what happens is that if the shear is low, it will deform a little bit, elastically, surely, then also flow a bit, but the displacement is not enough to break the network. If the shear stress is higher, then the non-covalent bonds between the polymer in the matrix will start deforming. And if the shear is very, very high, then the biofilm will basically flow and completely change its shape and structure. However, in this model, which is widely accepted in the literature, something that is still not considered is that in the exceeding a critical shear force regime, we are in a nonlinear deformation regime. And this, for example, leads to the formation to the streamers, but also could lead to the nonlinear behavior that I was mentioning before. And so active changes in the rheology of the biofilm. And this limited understanding of this important regime during removal could be something that has made our removal strategies less effective, or allowed biofilm to adapt very well to flow environment under very different conditions from the body to lakes to rivers. Okay, so we decided to study these behaviors, and we were really interested in studying these behaviors in biofilm formed under a flow. And now I will start from an experiment where we use a quasi-2D porous medium in a microfluidic chip, and we visualize the formation of the biofilm when we impose a constant flow and measure the pressure. This is our geometries. We have many pillars regularly staggered, and this is a picture of the microfluidic geometry after the experiment. All this white stuff that you can see here is biofilm. So in this case, it's a microfluidic experiment, but also at the micro scale, you can clearly see that it clogged. And now, if we zoom in, this is what happens. So first, we have an initial attachment, and then these filaments form the streamers. And then we start clogging. The black material is the biofilm growing. And then at some point, we have these white open channels that rearrange intermittently over time. And what interested us first was this flow path intermittency, where the channels form when the medium is fully clogged, but they don't stay in a single position and with a single width, but they keep changing and rearranging. Now, if we segment our geometry in biofilm, which is black, and channels that are white, and define a preferential flow path average width from these, we will see that we start from something where the channels are small, then at some point they become bigger, they open, we say, and then over time they narrow down again. And if now we plot the preferential flow path width as a function of the experimental time, and we also plot the corresponding pressure value that we measure with our sensor here, we see something very interesting. So when the preferential flow path become very narrow, we have a spike in pressure. And then instantaneously, very rapidly, the preferential flow path opens and the pressure decreases. And so we have this intermittency driven by builds up in pressure, because this builds up in pressure cause sloughing off of parts of the biofilm, as we can see here. These were like zoomed in, very zoomed in images of the biofilm. And this sloughing off allows the release of shear stresses in area where the shear is very high. At the same time, the biofilm compress. So in the region that are probably fluffier, when the pressure reaches a critical value, the biofilm can compress, and so allowing all the channel to become wider. And this drives the opening. And this is related to the capability of the biofilm to compress, to slough off, and this is basically the rheology of the biofilm. Conversely, the narrowing down is driven by growth. And we validated that doing an experiment where basically in one control we kept flowing nutrients and we kept having this intermittency opening and closing here. And in the other case, like in the channel close to it, we just flew some salt solution and nothing happened, because the biofilm without growth stayed in its open or narrower condition, but without changing over time. And if we now change the flow speed or the pillar size, we have cases in which this intermittency happens. Other cases here where it does not happen because the biofilm is too stiff or too fluffy. But by any means, we can say that the biofilm rheology governs the intermittency. Another case, examples of how the rheology can govern clogging, but this case at the pore scale, is in this simplified geometry where we just have a constriction that is much narrower than the channel. And in this case, we flow a suspension of bacteria. And we see that if we are above the yield stress of the biofilm, where the yield stress is defined as the stress at which above which the biofilm will become fluid, it will fluidize, and below it will behave as a solid. Well, when we are above, so it's fluid, it just forms a teeny tiny layer, while if we are below, it forms like a solid chunk that completely clogs the pores. And so here we could see in those two examples how really flow was shaping the clogging and the dynamics of this clogging over time. But here, in those examples, we could not probe the rheology, and we wanted to. And so we went to a much more simplified geometry. So we took a single pillar, and around this single pillar, we have a very big advantage, which is having reproducible growth of the streamers that grow in this case as two filaments suspended in the mid-plane of our channel. And this is due to the fact that we have recirculating vortices close to this pillar that accumulate biomass in the central plane, and then an extensional component of the flow that pulls this filament away. And this is what happens if we look at the pillar from like above. Those are our two filaments that form, and we can perform this experiment many times, and they will always form with the same geometry. As you can see here, we have full optical access, so we can use stains to probe the structure and the composition and see which matrix components are there and where they are located. But also we can use these images to reconstruct the 3D structure of our streamer and use a combined computational fluid dynamics simulation and particle tracking to reconstruct the 3D velocity field around the streamers. In this case, those two are the streamers seen like from here, from the front view. And this is the velocity measured by particle tracking and simulated. And as you can see, okay, experiments are a bit noisier, but in both cases, we reconstruct the same velocity field. But since we have to think, yes.
You're hearing?
Yes.
I just have a quick question. So the vortices that you're talking about here, they
Yes.
are the usual like vortex street behind the cylinder. So like the vorticity axis is not perpendicular to the model, but it's along the mean flow direction.
No, I mean normally the vortices that you're talking about would happen at much higher Reynolds number, like around 40. Here we are below 1, and they would be in the observation plane, so here. In this case, they are in Z, so along the optical axis, and they are kind of weak vortices because they are 1 order of magnitude weaker than 1 to 2 order of magnitude weaker than the main flow velocity. But this was demonstrated in the group of our stone around 2010. Those are vortices that form wherever you have a curved surface, and they form also at very low Reynolds number.
Okay, so this is not like an inertial vortices. This is
No.
like a viscous dominated. Right.
No, those are vortices that you can see emerging when you have at low Reynolds number a curved surface. And bigger the curvature, more important those vortices are.
And the curve here, the corner is between the cylinder and the top and bottom plane?
Yes.
Okay.
Yes.
Thanks.
Thank you. And so since, I mean, our streamers are like cylinders exposed to the flow, on them we will have a force creating then an axial stress if we consider just 1 liter slice of our streamers that is given by an hydrodynamic, the integral over the length of an hydrodynamic force where we have a pressure component and like flow shear components. These we obtain from the computational fluid dynamics for every streamer. And then this force is modulated by the shape of the streamer. And so once we know what is the local velocity and the wall-like structure of the streamer, we can measure the force or the axial stress in every point along the streamer. And this is very important because we can use the flow to probe the rheology. In particular, I mean, a rheometer, if we think about it, is just an instrument that imposes a force and measures a deformation. And we can do the same. So we have our streamers kept initially at constant flow velocity. Then At some point, we instantaneously increase the flow velocity and so the axial stress. And we can see, if we track the deformation in green, the elastic deformation and then the viscous deformation. And basically, this is like the textbook viscoelastic deformation that we had before. And knowing the force in each point of the streamer, we can calculate what we call a differential elastic modulus and the effective viscosity for our streamers. I mean, now we can characterize the structure, the rheology, the velocity field, and we decided to apply use this platform to characterize how the rheology and the morphology change when we vary the matrix composition and the ambient flow condition. We started by the matrix composition because that's pretty easy if you have mutants. So we used a model organism, Pseudomonas aeruginosa, that produces a biofilm with polysaccharides, PEL, and extracellular DNA. They have opposite charges, and so they are prone to interact. And using mutants, we varied the quantity of PEL from 0 to just a little bit and to like a lot of PEL. And first thing we could notice is that in all cases we form streamers. And so the polysaccharides component is not required to form streamers. Then we characterized the eDNA concentration, which was constant, and PEL, which was just a lot in the overproducer and then just a little bit in the wild type. And what we could see is that when we have PEL, the biofilm becomes stiffer. So in all cases, the biofilm is structurally stable, but it is simply stiffer when we have more polysaccharides. And this is an evidence that supports the fact that these two components can interact and create a stiffer networks when both are there. However, DNA is what really controls the formation and nucleation of the streamers, because if we just digest DNA with an enzyme, we do not form streamers on a single pillar, but also we prevent the clogging of porous media, because the streamer, like these very thin filaments, controls clogging of porous media. We also manipulated the DNA production using gene deletion, and also in this case we completely stopped the formation of the streamers. But also we could use this eDNA controlled streamer formation to promote streamers, make them bigger and bulkier when, thanks to an antibiotic, we induced eDNA release. And this is the case without and with antibiotic. This has been now medically studied because it can be very relevant when thinking about antibiotic treatments. Okay, so the take-home message here is that eDNA is very important and is the backbone of the streamers. And with this in mind, we moved on and studied the effect of ambient flow condition. We started with the same system of mutants, and we made sure that the bacterial flux was constant when we varied the velocity, because otherwise difference in morphology or rheology could just be done to the fact that we were accumulating more material. And when we looked at the rheology, we had a very big surprise, because what we could see is that we had a linear scaling between both the differential elastic modulus and the viscosity with the axial stress before the test. So biofilm grown, in this case they were independently grown, at each point is an independently grown streamers grown at a certain flow rate for like all night. And here what this graph is telling us is that stronger is the flow they see during their growth, stiffer they become. Literature would tell us that first thing, this is a long-term adaptation to local flow condition, and second, the prediction was that this would be related to a change in the matrix composition. But we could not detect any change in the matrix composition, density of PEL and eDNA were exact same. And so we said, okay, what's going on? Well, in polymer physics, this would be called stress hardening. However, in order for something like this to be called stress hardening, we need to grow a sample, in this case a streamer, at constant flow rate, and then test the same sample at low flow rate and high flow rate and see if we see the same changes in the rheological properties. And this is what we did, and this is what we saw. So the gray line is exactly the scaling that we found in the previous case of the long-term adaptation. And we see that our point, when we perform a rapid instantaneous switch, the points move along the same line, so they follow the same scaling. And here the data are divided based on the presence or absence of polysaccharides, showing that it doesn't matter if we have or not have the polysaccharides, our streamer behaves the same. And this then is a stress hardening by definition. And what is very powerful is that the same behavior is observed over long-term adaptation or short-term, meaning that this is a purely mechanical instantaneous response to pre-stress. We tested the same on different bacteria, different species, and in all cases we saw that they followed the same scaling when streamers were formed. And what this streamer had in common was the fact that eDNA formed the backbone of all streamers, as we can see here, because in eDNA is painted in red. And so we wanted to model the effect to better understand it, and this is what we did. We took a model for hardening that is very well known and studied in polymer physics, which is called the worm-like chain framework. And we said, okay, where do we start? Well, all these points from different species, different composition are scaling. The only thing that they have in common is DNA, so let's start by DNA. We estimated some parameters to describe the DNA network, and we simulated the same geometry and stress application that we had here. And this is what we found. Basically, we could get the scaling of our points. This is super important because the scaling is telling us which mechanism, which physical mechanism is at play. And the physical mechanism, which is like inside, like behind this scaling, is related to the entropic contribution from extracellular DNA. Basically, we have polymers that normally are coiled. We stretch them. We reduce the entropy when we stretch them a lot, and they don't want that for thermodynamic reasons, and so they become stiffer, and so stretching them becomes harder and harder, more stretched they are. This is nice. Still, we don't have a perfect agreement between the order of magnitude. And so this means that we are missing something in how the network is done. And this is something my group is currently looking into, because this constitutes a potential mechanism for biofilm mechanical resilience, both short-term, long-term adaptation to flow. And so we want to see how this happens exactly, how ubiquitous is this across morphologies, and if we can at all manipulate and alter this behavior. Okay, and this is like it for today for the rheology. Now, if you have any question, I will be happy to take them. Otherwise, I will move to the transport problems. Okay, so now, as we said before, the presence of the matrix also hinders transport. And well, transport in biofilm is quite peculiar and characteristics, because if now we think of our biofilm, the one that we had before, under shear not only deforms their structure, but will also like create a higher concentration of nutrients close to the surface of the biofilm, while nutrients or oxygen will have trouble penetrating the polymer network. And this is like an experiment that was done on this like sample of biofilm. Here they are using RTDP, oxygen sensitive dye, and in quiescent conditions, so without flow, we see that we have after a little while depletion of oxygen everywhere, while if we turn on the flow, the oxygen is repleted around the biofilm and depleted inside. If we increase the flow even more, oxygen is even more repleted around the biofilm and a little bit on the edge of the biofilm, where most likely is more fluffy, but is like stays depleted inside the biofilm, meaning that their diffusion and advection do not manage to spread out, to just reduce this gradient in transport and to reach the inner part of the biofilm. One big limitation of this type of experiment is that normally the sample geometry is like 3D and complex, as you can see here, and so difficult to study and model. And so in order to solve this problem, we created a microfluidic platform where we have a membrane made out of colloidal particles linked together. So this is like a solid membrane where we can tune basically the permeability and the porosity by changing the size of the colloids that we use. And if now we then seed bacteria on top of the membrane, we normally can, knowing the size of our bacteria, we can tune the size of the pore of the membrane and make sure that the bacteria stay mainly on top of it, and then give nutrients and promote the growth of the biofilm. The biofilm here is like this fluffy green material that is growing on top of the membrane, which is marked in white. And now then, if we look like from the top, we will see our biofilm front. And in Z, the channel is thick enough to have a biofilm, so many cell layers, but thin enough for the biofilm to occupy all the channel and be like a homogeneous slab. And the transport happens from above the biofilm, like from here, and so we can expect a gradient of nutrient and oxygen forming to the interface of the biofilm inside. And this is what we saw. We probed, as in the previous example, the oxygen gradient. And this is our biofilm here. This is the biofilm that grows over time. Initially was smaller, then over time it grew. And if we visualize with the same dye the oxygen concentration, we see that on the top here we have high oxygen, and close to the membrane, so inside the biofilm, the oxygen is much lower. And this is how the gradient, that is in this case represented by the intensity of fluorescence of our dye, evolves over time. So over time, we have like brighter biofilm close to the membrane, which correspond to more and more depleted in oxygen. And we can see that this has like consequences on where the biofilm grows, because now here we are like using, this is the image of the biofilm growing. You will see it starting soon. And here we use optical flow analysis to basically see where the biofilm is expanding. And we can see that the brighter color, so the higher expansion, happens very close to the surface of the biofilm, while the biofilm in the inner part basically is like moving way less. And now if we plot like profiles of expansion rate versus the thickness, we see that at 0, so 0 is our interface of the biofilm is expanding a lot. And then at the membrane surface, basically, the biofilm is not expanding anymore, and there is an intermediate region where the biofilm expands very, very little. This will reflect also difference not only in transport, as we saw before, but also in metabolic activity. And this reduced permeability then and differences in metabolic activity that it induces will not only limit solute penetration, but also limit the susceptibility to chemicals, for example, to antibiotics, leading to reduced treatment response inside the biofilm. And this is something that we are studying now, and I'm not showing the images like in the sake of time. Because what I want to tell you something about before finishing my talk is like why this happens and why this develops over time with a bit more mechanistic explanation. So the approach that we are currently taking is the following. We have our biofilm that we can beautifully visualize, and we measure the thickness of the biofilm over time. And we have sensors in our platform that allow us monitoring the flow rate exiting the channel when we keep the pressure at the inlet constant. And as we can see, in the black case, we just have the membrane. Basically, we can maintain a constant flow. When the biofilm is growing, the flow rate measured drops over time. And then from like we have the thickness, we know the pressure, we measure the velocity. So then we can infer what is the hydraulic resistance of the biofilm or the permeability that we prefer because it's normalized, is more like easier to link it to a material properties normalized for the biofilm thickness. And in this case, what we see is pretty striking because thicker the biofilm becomes, so correspondingly older the biofilm becomes, less permeable it becomes. And this is per unit length, so it's not related to the thickness, it's related to the biofilm properties, meaning that biofilm maturation is progressively suppressing transport in the overall biofilm, because this is a number for the whole slab, no maps here. This can be looked at in different species. So here we took some commensal and some pathogenic strains, and we measured them in the early stage or in the mature stage. In the early stage, we don't have so many significant differences, while in mature biofilm, we see like a clustering, because these 4 species, which are model organism former, model biofilm former, but also pathogen, exhibit a statistically significant difference permeability from their commensal counterparts. And this is something related to biofilm maturation, and is also very interestingly a pathogen-specific reduction in permeability. So this is something that we are currently studying. However, could like give very good hints on why pathogens are so well resistant in case of antibiotic treatments and chemical treatments. So, but now we were talking about biofilm maturation, right? And when a biofilm matures, what the difference is that they are producing polysaccharides, proteins, and DNA. And so in order to disentangle the role of these different components, we took one model organism, S. epidermidis, and then we used the deletion mutants for the structural protein and the polysaccharide. We grew the biofilm in all cases. And what we saw is that while in early stage they are all similar, early stage basically they are all packed cell, so we don't see a big difference because the cells are the same in all cases. But in mature biofilm, where in the wild type, so the biofilm where we have all the components, the polysaccharides production and the protein production and the kicks in these like biofilm are much less permeable than, for example, the biofilms where we don't have the polysaccharides. And so in this case, what we could see is that the polysaccharides, if we delete them, we can restore the biofilm permeability even when biofilms are mature. This was not only the case for epidermidis, but was also the case for Bacillus subtilis, another strain where the delta matrix and the delta EPSO, these two are slightly different mutants that are not able to produce the polysaccharide, were way more permeable than the wild type. And also in E. coli, another biofilm former, were same. When we did not have the polysaccharide here, we had like twice the permeability than in the wild type case. And so this is like something very interesting, but we wanted to understand even deeper and see if we could rationalize this. And we adapted a model that extended the transport theory from synthetic gel to biofilm. And in this case, we treated the cells as impermeable inclusions and the matrix polymers as cylindrical obstacles. Since those components, those are model organisms, and the size of these components is known, we could benchmark the size of the matrix polymers and the cells in our model. And then we were also able to capture the EPS heterogeneity through a network with some level of clumping. And basically what we could see was that we could model reasonably well the permeability in terms of real numbers, real parameter of the wild type biofilm, but also by assuming a 90% reduction in the polymer production, we could capture the increase in permeability in the delta polysaccharides and delta protein. Like this is pretty insightful because, again, not only for the mechanical response we could rationalize everything in terms of a polymer network, but also in terms of transport, and say that in one case it was DNA dominated, in this case the transport is polysaccharide dominated, but in both cases are like the polymers that constitute the biofilm that confer their characteristic properties. And so moving on, we want to more and more try to see to which extent biofilm can be described as living polymer network and analyze and rationalize their properties in terms of network properties, looking really into specifically the crosslinks, the EPS interaction, the packing fraction, and also see how does the network respond to chemical stressors, polymers additions, ions, pH. And with that, I am at the end of my talk, and I would like again to thank the people that did the work, you for your attention, all the international collaborator and funding agency, and I will be happy to take questions.
Anybody has some questions from Trondheim?
So can I ask a question on the biofilm, especially deep inside? If you have specific enzymes that may cut the DNA in certain places that are just really tailored to the DNA, extracellular DNA, can you see a difference, or did you try to loosen up that or destroy the biofilm?
So we did in case of streamers, and if we add DNAs, streamers are not formed. And these are the data that I've shown where we prevented basically the clogging. And so there, I mean, is formed or not formed. We had the streamers or not had the streamers, and so we could not probe the mechanical differences in one case or the other. In case of the transport, we did not try to make this treatment yet. What we can see, and is related to this phase where the enzyme are produced, that is the dispersal phase, on the membrane, we can see that the swimming, the bacteria that are swimming, there are like waves of dispersal. So when the biofilm is mature, there are moments where cells swim away from the biofilm. But normally, based on the resolution we have, this is limited to the outer layer. We do not see cell escaping from the inner layer of the biofilm. So if this is the surface, they escape from the surface.
Okay, thanks. Can I ask?
Yes.
Okay, very interesting talk. I have one question, because you talk much about the hydrodynamics, but of course there's also a lot of biology, and I'm not a biologist. But one thing I was wondering about is, normally, if there's free DNA outside cells, the immune system recognizes it and attacks it. So what's happening in this case? I mean, are these biofilms triggering an immune response as such?
This is a very interesting question, and is something that we will look into in the coming years, also in collaboration with the University Hospital in Zurich. So, what clinicians see more as a problem is actually the opposite. So, when we have an inflammation, we release more DNA, because our cells like leak DNA or break and release DNA, or there is a defense mechanism.
Sorry, can I just interrupt? The DNA that is the core of these streamers, that is the eukaryotic DNA from the cells of the victim, and not the DNA from the bacteria.
No. Now is the one from the bacteria.
Oh.
But since DNA assembles mainly due to hydrodynamic driven assembly, I did not present this data, but we have like a toy model published in the same paper where we talk about the stress hardening. So we basically have shown that the assembly of the streamer is driven by the fact that DNA chains assemble in flow. And this is not like related strictly. So the cells in this case are just a source of DNA. And this DNA comes from the lysis of, so basically the explosion of a few cells in the populations. But DNA is so big that even if just a few % of cells in the population die and lyse, and this is something that is physiological in healthy bacterial population, well, that traces of DNA, being DNA so big, are enough to trigger the assembly, especially in flow, where basically DNA is like stretched, and so the linking sites of the molecule are like exposed, and so it becomes more sticky. And this is something that people studying single DNA molecules have already seen and shown in single DNA molecules. Here our DNA is a bit bigger and a bit more complicated because it comes from the lysis of the cells. But our model suggests that the assembly mechanism is the same. And this, in principle, could also be extended then to the human DNA. So if we have an inflammation, or if in a river some fish release DNA, well, that DNA could be very well incorporated in principle into the biofilm, and so contribute to the growth of the biofilm and to the stiffening of the biofilm. And so, yes, we have a lot of biology, but in this case is like more polymer physics related, both the formation and the incorporation of the DNA.
Okay, thank you very much.
I have a question that might be, well, you both, because I don't know if I have enough background on biology to ask this. But provided that, let's imagine this biofilm as its own thing, is a composite of a polymer matrix with some bacteria, right? And then you have a flow with nutrients where you allow the system to grow. You've shown that as you increase the shear, there's like this shear hardening, and you also show that the oxygen concentration increases as you grow, as you increase the shear, right?
Yes.
So does that mean that the behavior of the living matter inside this system is linked to this behavior response of shear hardening? Like the bacteria transporting inside this bulk system, could they be influencing the mechanical behavior of the system?
So this is a very interesting question with a very complicated hypothetical answer, let's say, because the honest answer is I don't know, because it should be studied. But there are things that we know that could lead to hypotheses. So the hardening that we have seen in DNA-rich biofilm that are the streamers, so we don't know how general this is, but in the streamer this is true, is just a polymer physics phenomenon. Then what is the advantage for the bacterial population? So imagine that we have like two chunks of bacteria that are here, they are close together and they are interacting. If the biofilm was not hardening, I would pull the streamer and they would just drift away a lot. And if they were interacting, like exchanges things, as in the city of microbes vision of the biofilm, they would completely lose this interaction. And it would be very bad because sometimes in biofilm, different parts of the biofilm are interdependent, like a bit in our body, like some behave as a stomach and some behave as lungs, and so perform different function. And if by drifting apart, they would suffer. But if the matrix that keeps them together is hardening, well, the level of hardening that we have seen tells us that they will deform by 10% of the length of the streamer at most. And so just they drift apart a bit, but not too much. And so somehow they can remain in proximity, and maybe they will have to adapt their interaction, but they will not completely lose it. And so is this on purpose? Is this an advantage? We don't know. However, having a material that keeps you together as a community could be a big advantage if we would imagine the biofilm, and this is something that has been validated multiple times. If we imagine the biofilm as a city, as a body with organs, as like an entity where the individuals find benefit in a mutual interaction.
Okay.
So I hope it was kind of sort of clear.
Yes. Open more questions, but yes.
Yes. I fully agree, but yes.
Any questions from Oslo?
I have one here. Thanks a lot again, Eleonora. I was just wondering, so two practical things. Do you have any way to reuse the models, like when the biofilm clog the whole thing? So you can't really get rid of it, right?
No, it's our models are just PDMS. It's a silicon, it's very cheap. So it would be more expensive to try and clean them than trash them.
Right.
But I can see that if the model is, for example, a rock that you carved, how you would like to reuse that.
Right, yeah. And how do you avoid the biofilm from clogging places that you don't want to clog, like the outlets of the model or the ways towards pressure sensors, all of that?
So it depends on the geometry. So in the streamers platform, the clogging wasn't much because the cells were growing very slow. And so everything remained pretty clean. In the membrane device, the one where we have the membrane and the biofilm grows on top, the biofilm, since we see the very few cells and we flow them very fast on the membrane, we can confine them there and avoid the clogging in the inlet and at the outlet. But because there we really need a super minor number of cells, and for the cells we have a dedicated inlet, and so we keep the rest of the geometry relatively clean. And so that basically in those two cases, in the streamers and here, we were like carefully placing inlets to limit the colonization. In the experiments that was with the 2D porous media, there was clearly more complicated. And there we had growth of the biofilm in the region before the porous media, like we had a triangle before the pillars, and there we had biofilms, and we couldn't just, we had to accept it and have bacteria there. However, we managed to avoid clogging in the pipes by seeding the bacteria and then putting a clean tube.
Right. And just to finish, so you mentioned a technique called optical flow analysis.
Yes.
What's that?
Basically, I mean, now is something, if you look for optical flow algorithm, you will find many. And those are basically techniques based on correlation of images. So instead of, when you do particle tracking, you have algorithms in which you correlate the position of a particle that is here at T1 with the particle that is there at T2, and there you correlate the position of objects. Well, if you use an optical flow, you have to imagine that instead of having a distinct object, you have like a carpet that is deformed or is flowing, but some features that are in the carpet are still there at T1 and T2. And so basically an algorithm that is based on time correlation of images will see a certain level of correlation and basically measure the displacement of these features, even if it's just like a carpet-like pattern and not individual objects.
Right. Super. Thank you. More questions here? No more questions from Oslo?
Yes, I think we're good here.
Okay, so thanks a lot
Yes,
for the lively discussion.